Started with purine synthesis. So we're going to zoom out a little bit. Where the heck are purines coming from? Why do we care? What is a purine? So let's start with, what is a purine? So, what are some answers to that question? What is a purine?
What is it?
It's a two-ring structure
Good.
that contains some
Carbon, nitrogen.
Yes, carbon and nitrogen and rings. Yes, it's a two-ring structure, and it is used to make DNA, RNA. It's a nucleotide. Okay? So purines are nucleotides, and so I like to remember purines with a little phrase. Purines have adenosine and guanine, and so I like to remember all girls are pure.
So true.
Yes.
There we go. So all girls are pure, and then for the pyrimidines, which we are not going to be talking about in depth today, but pyrimidines are the other two nucleotides. And for those, I say, you see the pyramids. So you can use that phrase either to remember pyrimidines or if you're going to become a tour guide in Egypt. Either way works. So today we're going to go over the purines. Now, a nucleotide is going to be a sugar, typically ribose or deoxyribose, deoxyribonucleic acid. So we have a sugar as one part of a nucleotide. And then we have a base. And the base is either going to be adenine, guanine, uracil, or
Tyrosine. No, not tyrosine. It's the other T.
there should be a T up there. I think
There
there we go.
What is the T?
Oh, there's the. Okay, you see the pyramids. All right. I'm like, where did my thing go? All right.
Oh, because you did RNA versus DNA with the U.
Yeah.
The U. Okay.
Yeah. So you stick any of those bases here as one of the things, a sugar and a phosphate. And so those three things make up a nucleotide. Now, there's also something called a nucleoside when it isn't completely put together. And so we just take the phosphate off. So that would be this guy off to the side, usually coming off of the 5th carbon. You can have a phosphate there, or you can take it off. If there is no phosphate, then it is a nucleoside. So, same thing, but no phosphate. Okay, and so these are the guys that are used to make DNA and RNA, and they're also used for a lot of energy carriage throughout the cell. So they're energy storing. What is a molecule that we are very familiar with, but we may or may not be seeing it as a nucleotide, usually when we look at it, like thinking about it that way? ATP. Excellent. ATP is a nucleotide, except it goes, it has more phosphates attached. So, all right, excellent. So now that we've gotten kind of the idea of what the heck is nucleic acid, what the heck is a nucleotide, a nucleoside, purines, and pyrimidines, let's go ahead and see how we make purines. All right, so we go back to our very favorite pathway, glycolysis. Our first experience with trauma. All right. So coming off of glycolysis at G6P, glucose 6-phosphate, we have two pathways. One of them is Glyco Genesis, where we're making glycogen. Not talking about that one too much today, but going off also off of glucose 6-phosphate, we have another pathway. What is that pathway?
PPP.
PPP. Excellent. So PPP is going to come off, and then it would come back and join around the fructose area. But we make a product right around here, and that product is ribose 5-phosphate. Okay? And so we take glucose, and we can either make it into ATP energy down here by recharging ATP, or we can send it off this way to make new nucleic acids. So how do we go from ribose 5-phosphate to purines? Thankfully, it isn't too many steps. So we are going to make something called PRPP, and we're going to use an enzyme that is named after making that molecule. So when we're making a molecule, we often call them synthases or synthetases. So this one's a synthetase.
So it uses
Yes,
ATP.
synthetase uses ATP. And so this is going to be PRPP synthetase. Awesome. And conveniently, the enzyme on the other side is also named after PRPP. So it's PRPP, and in this case, it's amidotransferase with a D, amidotransferase. PRPP, amidotransferase. Okay.
Wait, what does the amido mean?
Amido, so an amide group looks slightly different than an amine group.
Amide.
So it's an amide,
Yeah.
if you know it, yeah. So it's transferring an amide group. Okay, and so this is going to eventually make something that I'm going to bend down around here just so that we can have room to write about it. IMP. The I stands for inosine.
Is it that important for us to know that intermediate between those two?
I don't recall it being important to the point that I don't recall what the intermediate was. What was it?
5-phospho-beta-ribose.
I don't recall that being something that you need to remember. It isn't involved in any disease processes. These two enzymes are going to show up in diseases. So I will make this a dotted line so that there is an intermediate in there somewhere. All right. So, pretty simple so far. PPP to ribose 5-phosphate, ribose 5-phosphate to PRPP using PRPP synthetase, and then our second PRPP is PRP amidotransferase. So there's two PRPP enzymes. So go ahead and review that in your head, and just sandwich the two enzymes around the PRPP, and then it should be pretty straightforward. Just make sure you got this, because it can disappear, and then it's like, where the heck do these come from? All right. So, now, really quickly, before we write out a large thing over here, this is all, so far, de novo, meaning making it from scratch, new, novo meaning new, de novo synthesis. So we are making these purines from nothing. Well, from glucose, but from nothing. We're not taking them from anything that was a nucleic acid. So we call that making brand new nucleic acids, or in Latin, de novo synthesis. Now, as we get into this next part, where we have a lot of dancing between, there's a whole bunch of nucleotides, and we're going to see this next part is focused on salvage. And so this part's de novo. Now we're moving into salvage. And so salvage will be the recycling part. They kind of have a mix right in the middle where you're kind of doing a bit of both. So we are going to go ahead and draw out like this. AMP, IMP, GMP. So this is adenosine monophosphate, inosine monophosphate, guanosine monophosphate. Cool? Okay? And adenosine monophosphate can be broken down to just adenosine. Adenosine. Inosine monophosphate can go down to inosine. We're just taking the phosphates off. And guanosine monophosphate can go to guanosine. Okay? So all we've done is knock the phosphate off. And so then it's not monophosphate anymore, it is just itself. Okay? Now, going down one further, so right now these are nucleosides, right? No phosphate.
Yeah.
That's all we did. We went from nucleotide to nucleoside. No phosphate. Now we're going to go from nucleoside down to just the base. The base. All right. So,
Guanine, adenine.
adenine. Yes. Adenine. There's a letter missing in here. The letter S is gone, okay? There. And so the way that I like to remember what is happening here, the sugar is gone, the S is gone, okay? So we are just at the base. Adenosine still has a sugar, ribose. Adenine has no sugar. It has lost its letter S. It is all alone, on its own, flying free. All right.
Lost its sweetness.
It has lost its sweetness. We still care, though. All right.
Add some stevia.
Oh, so harsh. Oh. All right. Don't listen. Okay. Inosine.
Inonine.
Right? It's like, how are you going to do that? Well, we don't. We make this one into hypoxanthine. Okay? And hypoxanthine, inosine's already weird because it's like, who's ever heard of it? So hypoxanthine. So the one in the middle is going to be weird. You did, however, take the sugar off. Now, over here, we're going to get guanine. Exactly. Okay? So, this is a process that we would do we'll get to that in a second. It makes sense when I draw a few more things. But I want to pause right here, try and remember the three that we're dealing with and how they break down. So go ahead and shoot for it. Nucleotide, nucleoside, base. And then what does that actually mean in like what are the molecules? So list out the nucleotides at the top. And then when you're ready, the nucleosides in the middle. And then the bases at the bottom when you're ready. All right, so at the top we had A, I, and G, MP. And then in the middle we had adenosine, inosine, guanosine with the S's. And then at the bottom we just took the S's out and then we had a weird hypoxanthine in the middle. All right, so now we're going to finish the drain because this is the way that you would break them down to get rid of them. Okay? So we're going to finish getting rid of them, and then we'll show you how to say, no, wait, we need those, and rescue them. Okay? So, really quickly, I would like to throw in.
Is it salvage where the rescue is?
Yes.
Like salvage.
Salvage is rescue, same thing. Alright, so here is an enzyme in between these two that I want you to know for breakdown as well as for salvage. Alright, ADA, adenosine, is where the AD comes from, and then the A is like transaminase or something like that. I figured I'd remember exactly what it was, now it's just ADA. Okay, so ADA will change adenosine into inosine, and we want that to happen because we are good at dealing with hypoxanthine. Okay, so when we're breaking down, we're trying to get these two guys to end up as hypoxanthine. Cool? And then guanine is left all alone by itself on the side, and it's going to do its own thing because we're good there. Alright, so these are both going to be handled with the same enzyme to create the same product. Hypoxanthine and guanine are going to be acted on by xanthine. We saw hypoxanthine. Now we're going to use an enzyme called xanthine oxidase. And xanthine oxidase is going to act on both sides, so I'll just draw a red arrow to each of them to just denote that that is happening in both places. And we're going to make xanthine. And then once we have xanthine, we go ahead and use xanthine oxidase again to oxidize the molecule a second time, which gives us uric acid. Okay? So from our previous chunk where we had hypoxanthine, it's like hypoxanthine, xanthine, uric acid. Oh yeah, guanine comes along for the ride. And then the enzymes are both, the enzyme is named after xanthine, and then up here you have one that's named after the adenosine that we're trying to shunt over.
So guanine can be xanthine as well?
Yes, guanine will turn into xanthine without needing to go through inosine. It's its own thing off to the side. It's kind of like there's an invisible line between these two, where it's like guanine does its thing, adenosine and inosine work with each other, and then everybody ends up at xanthine and then uric acid.
And then the adenine, what's going on with him? I think that there.
Adenine, this is to show that you can make adenine, but if we're breaking it down, we typically will shift it over to inosine and hypoxanthine. Adenine, if you build up some of it, I believe it can give you issues. I also believe that you can want a base to go and form some other molecules. Nothing that we're concerned about, except for noticing that this is where it comes from.
Okay.
Any other questions?
What's the positive regulator that takes glucose and makes into ribose 5-phosphate?
So we have regulation, this is an excellent question, where if we don't have very many purines, then that makes so that these two enzymes, PRPP synthetase and PRPP amidotransferase, those are so if you have a whole bunch of purines, let me put it this way, if you have a whole bunch of purines, then these will repress those enzymes.
Inhibitors.
Yes, so they will inhibit. And so instead of drawing like super messy lines all over the place, the mess. I'm just going to draw a bracket off to the side and say purines. It says purines. And that inhibits, and I'll put the inhibit sign. That inhibits those two. And so if those aren't inhibited, then they will move forward and you will get it. And so if you're low on these, you'll make more. If you're high on these, you will inhibit. because you'll have a whole bunch of these molecules bumping into those enzymes and shutting them off. Make sense? Excellent question.
I have another one.
Please do.
So, what's the whole purpose of making all this if the uric acid is going to be excreted out? Like, is there...
So, we also, without going into, we're used to seeing ATP, ADP, and those are, once you have AMP, you can then make ADP and ATP through pathways that we don't need to know exactly. It's just a phosphorylation. You add a phosphate.
Okay.
And so you would just change AMP and GMP into ATP and GTP, or whatever they're needed for. Or maybe you're sticking them in the DNA, and so then you would put them in that form, and that's not going to be TP. That's just like, okay, we got it, let's stick it in. So AMP in that case. So whatever you need, you'd make. Question?
If we have a lot of purines, we'll shut them off with two enzymes.
Exactly. If we have a lot of purines, if we have a lot of purines, we will shut off the two enzymes that make them.
And does the adenosine ever become adenine?
Adenosine becoming adenine? Yes, it can. There are two ways it can go. So typically we see adenosine going to inosine, and then it just follows the inosine pathway down. But you can have adenosine become adenine, especially if this enzyme is broken. There are 5 diseases we're going to talk about today. One of them is, I call it SCIDS, but SCID, severe combined immunodeficiency. So really bad, you don't have an immune system disease. When you don't have this, then you end up getting a freaking ton of adenine and adenosine buildup in your immune cells. And that makes so that if you have a freaking ton of these guys, they all together, all of these guys would come up. We're jumping ahead, but I like that. I like the question. So, all right. They would come up. You have a whole bunch of adenosine, adenine, and AMP because you can't change it to inosine, so the whole thing backs up. That means AMP is high, and that means these guys get inhibited. And so your immune cells, especially, because they do a whole lot of nucleic acid work, and so they're very sensitive to this deficiency. Even though GMP is super low, you have so much AMP that it still shuts it down. And then they have no GMP and no synthesis. And so then your immune cells don't function super well. All right. So that's kind of messy, but yeah, so, and we will revisit that disease. Question?
Yeah, can you explain how, like, guanine can feedback and go to INP and all of that?
Yes, and that is just where we were headed, so excellent. And I'm going to get rid of these lines right here because they were for a disease, and I would like to do diseases in a different color so it's not super confusing. And we will get to those. All right, so we just did metabolism. And that's when you break it all the way down and uric acid, then uric acid goes into the bloodstream and eventually you would pee it out. Really quick, before I get to another question, I would like to show the salvage, which is what happens to recycle these guys. So if we make it and then we break it down, it would be really cool if we could use it again because it's only a sugar and a phosphate that need to be added. So we are going to use, I'm going to use blue for this because these enzymes are awesome. All right, we're going to use HGPRT. I call it Higpert. It's just a lot easier. All right, Higpert and Apert. Now, the PRT, really quick, so it's like, what the heck is PERT? Well, besides, someone who's PERT, like, are you being PERT? If your grandma said that. All right, so the A and the H and the G, those are standing for the the bases that they're dealing with, okay? The P is standing for a phosphate, so we have adenine, phospho, ribo, transferase. The ribo is referring to ribose, so we're adding a ribose sugar and a phosphate. Phosphate, ribo, and we're transferring them from something else and putting it on there. So that's why it's called PRT, is because you are transferring a phosphate and a ribose back onto adenosine, guanine, or hypoxanthine. And so that means that these guys, using that enzyme, get to be recycled. Okay? And then the hypoxanthine also follows that recycling pattern. All right. So that right there, with those two enzymes added, that is salvage. Okay. So, backing up just a bit, to keep it straight in my head at least, I like to think about metabolism as one thing and run through metabolism. Once I'm done with metabolism, then I will turn that part of my brain off, and then I will go ahead and look at salvage. And it's like these two things go up. So go ahead and see if you can remember the bottom bit of metabolism. We make the three things, and we've done that. And then we are breaking it down from the bases into xanthine and uric acid. And go ahead and see if you can go through that and then see where those are on the chart. And so you can say right there is this, right there is this, right there is this, and then they go down to this, down to that. Hopefully that was enough time, and if not, it was adenine, hypoxanthine, guanine, all going down to xanthine, and then xanthine going down to uric acid. And the enzyme being used is xanthine oxidase, twice. All right, so, and then salvage. Go ahead and close your eyes and see in your minds where the salvage enzymes were and what they were named and why they were named what they were named, at least for the bits at the beginning, so you know which ones deal with which bases. All right, so we had HIG-PERT and A-PERT. The first letters besides PERT are the bases that they deal with. All right, awesome. So now that we've gotten up to that point, did we have any more questions?
You answered my question.
Yeah? Okay, awesome. Any other questions?
It's still bothering me. Why we need ADA? I know that it helps to convert the adenosine to guanine, but why the body does that?
Why do we need ADA?
I know what it does.
Like, why doesn't adenine just
Yeah.
go down? That's a great question. I can only give a guess. I don't know on this. My guess is, there's two guesses. One is maybe adenine, just biochemically, is difficult to change into xanthine. Or perhaps it's so that they have fewer, you see how HIG-PERT right here is taking both the bases that turn into xanthine? That way you only need one. And so you can say, all right, when we want to have less xanthine and uric acid, we'll use HIG-PERT especially. And over here, adenine is used for ATP all over the place, a lot. And so maybe because adenine is used for different products, it's off to the side, so it can be regulated differently. But again, that's a guess. It's probably so that the body can regulate it differently. So,
I have another question.
Please.
So I know I think last time I made a big deal about one of the enzymes. It was the Purine nucleoside phosphorylase.
Okay.
Is that important to include there, or should I not even think about it?
Purine nucleoside phosphorylase.
It's the one that makes inosine go to hypoxanthine and guanosine go to guanine. So it's like more specific, but they called it this generalized name. So instead of nucleoside, it would be inosine phosphorylase or guanosine phosphorylase.
I believe that those ones, so the enzymes that I'm not putting in, obviously all these arrows has enzymes next to them.
Right.
I don't think you need to memorize them, but for your information, all of these, to my memory, are phosphorylases because we are taking a phosphate off and then we're throwing it into the cytosol, not onto anything. So it's a phosphorylase because that's when the phosphate's just floating through the water rather than being put on another molecule. Inorganic phosphate. Anyway, the point, you can dump all of that. That was geek. So, phosphorylase. It takes off a phosphate. And then these ones that take off a sugar, those are kinases for phosphates when you do stick it onto a molecule. We use phosphorylases for the first bit. This next one,
I think it's also phosphorylase.
I don't remember. If anyone double checks, Pedro thinks it's
nucleoside phosphorylase.
Phosphorylase?
Yeah.
Says it there? Okay. Cool. But those ones are not ones that she expects you to remember. She wants you to know the ones that I've written in red and blue, and because those are disease related.
So can you clarify for me, Taylor? We're using the APRT and the HGPRT to, I mean, those are our salvage enzymes.
Yes.
But why wouldn't, we're salvaging to go back and make AMP to then go up and make ATP. Why wouldn't the body just, for AMP, just keep it as AMP instead of breaking it down to adenine and then?
That is a very good question. And the answer to that is, I don't know. So I would have to look in more to what adenine is used for versus AMP going to, because I'm inferring the AMP goes to ADP goes to ATP. So yeah, I do not know. The only thing that I've ever been able to come up with is maybe there are some processes. You know how it costs an ATP occasionally? And at least in beta oxidation, when the fat first comes into the liver cell and you're putting a CoA on it, and it makes acyl-CoA synthetase to make acyl-CoA. That takes 2 phosphates off of the ATP. And so it goes from ATP down to AMP in a single reaction. My guess is that there are some reactions that go beyond the monophosphate, and then it's just like...
bring it back to the monophosphate.
Yeah, because once you've gone down here, you can't just bump up for free.
Right.
And so you have to go down to get the recycling. And... that, or you can use AMP the same way as ATP, and all the textbooks say ATP because it's like we have to pick one. And so you just could use AMP instead of ATP for all those reactions, but then that means you bump it down to here, and it's whichever adenosine, tri, di, or monophosphate bumps into it first, and then it would get reduced or oxidized, dephosphorylated down to adenosine. Then you have to go through the rest of the way just because it's not chemically favorable.
Okay.
Does that make sense?
That does make sense. I guess my only follow-up question would then be, so am I understanding this right? If I have guanine, is it based on feedback mechanism? So whether it's going to go to xanthine or not. So if I get down to guanine and I don't need to excrete anything as uric acid, then I'm going to use PIGPERT to go back up to GMP.
Exactly, it's feedback.
Okay.
What does the body need? We have enough, let it go. We don't have enough, bring it back.
Okay.
And so when these guys are running, typically you will have these guys running. Because these are make it new, these are make it from old. Both of them are make it. And so that means that they're going to be regulated similarly. And so if you have a freaking ton of these, then it turns these off, and it would turn these off as well.
Okay. That makes more sense.
When you were geeking out, and you said that, will you say what you said again and then tell me what it's called when it does actually put the phosphate on something else in the cytosol?
Okay, we're going there.
Sorry.
So, we have phosphorlases.
Yeah.
and we have kinases.
Yeah.
And they both deal with phosphates, and that's normally all that people would care about. But I went and read on things, and most of the time, almost all the time, if you get deep enough into naming stuff in science, there's always things that people named wrong, and then they never fixed it, and so it doesn't always work. But in general, phosphorylases are going to take a molecule. I'm just going to use ATP as an example because it's definitely got a phosphate. If we took the phosphate off of ATP and we cut it off and made it ADP plus inorganic, that's the lowercase I, phosphate. Inorganic phosphate means it is not bound to any kind of organic molecule. It's free floating. It's like a phosphate, which looks like this. Just floating off on its own. Kinases, on the other hand, will take ATP or whatever, and they will make it ADP and some other molecule, and so it'd be ATP plus.
So they throw it on something else.
Yeah, they throw it on something else. Let's just say glucose, I guess. And then it would be glucose 6-phosphate. And so they actually throw it on something else. So that's the difference. So if it says kinase, then 99% of the time it is throwing it onto something else. If it's a phosphorylase, 99% of the time it's throwing it free into the fluid.
Cool.
So yeah, it's like, finally, okay, why, okay. And yeah, so if she ever has multiple choice and she has inorganic phosphate listed as a byproduct in one of the answers, because she'll never ask you this straight up, but it's like, I know that it's a phosphorylase, therefore the answer that says inorganic phosphate has to be right. That's how we'll pretend that this was extremely applicable. All right. Awesome, I love it. Makes me happy. All right, so any other questions? Okay, then, yes, the diseases. All right, this is where we get to jack the body up. So, let's start with, we're going to start with gout, but I'm trying to pick out a color that is unique.
I have a green one.
Looks like this green is okay, but I may take you up on that in the future. All right. So, 5 diseases. All right? They're not too bad, in my opinion. Uric acid. This is a disease. That when you have too much uric acid. What disease do you get? Gout. This is one that somebody's ancestor has had at some point. Your parents, your grandparents, maybe yourself. You're your own ancestor because you're that wise. All right. So this is gout, when you have a freaking ton of uric acid. Now, why would we have bless you, why would we have, I've been with Dr. Nick too long, I almost said it twice. All right, so gout, why would we have too much uric acid? Answer, there are 3 things, all right? If this guy is going too fast, if we've got too much synthetase and it keeps going no matter what, okay? So number 1 is synthetase. And I'm just going to put numbers down here, and all right, synthetase. 1 and 2 are both around the PRPP, is how I like to remember it. 1 and 2, both the PRP enzymes. This guy right here is not being inhibited.
What did you put in the asterisk? What's that asterisk? Arrow. Oh, okay.
Next to the asterisk is an arrow meaning up, meaning it's going too much.
That was a 4. Thank you.
It's going 4 times too fast. That's not true. Okay. So yeah, both of those are going too much. Why is that a problem? We're going to make too many, and that means we have too many, and that means we don't need them, therefore we break them down. So we made a whole bunch of nucleic acid, and then we just broke it all down because it's useless, because we have enough, and that means lots of uric acid, that means gout. Now what's the other way that we could get a whole bunch of gout? is if we got rid of especially Higpert. Why not Apert? Who can tell me why not Apert?
Because it doesn't make uric acid.
Exactly. Perfect. Apert does not produce xanthine. So Higpert is the number 3 for gout. So 2 PRPPs and then Higpert. They all start with P. It's great. All right. PR. Okay, that's not going to be legible to anyone. I'm going to get rid of the synthetase and replace it with just PRPP. PRPP enzymes, and then Higpert. Okay, so that is gout. And if we wanted to treat gout, one second, there is one other thing that we could do, and that is throw in something that is called allo, meaning other, purinol. So other purine. It's not a real purine, but it looks a lot like a purine. And so aloe makes me think of the aloe plant, which I am drawing right here. All right, that's totally an aloe plant. Anyway, so allopurinol means that you have something that looks a lot like some purines, and that tells, I believe it looks a lot like xanthine, except it can't be broken down. And so the xanthine oxidase is trying to break down the allopurinol.
It looks like hypoxanthine.
Thank you. Hypoxanthine makes more sense because actually it couldn't go backwards from there, so that wouldn't work. All right. Thanks. Always double check me. All right, so hypoxanthine. And so if these enzymes, xanthine oxidase is trying to break allopurinol down and it can't, well, it's busy, so it can't break down the other guys, so that slows down the uric acid production. Make sense? These guys are busy with this allopurinol, therefore they can't do their job of making more uric acid. And so that's what we would do to treat gout. Well, one way. All right, so, question?
I just wanted to ask, so if the 2 PRPs are like hyperactive, right, then you get gout, and if the H2PRT is like inactive?
Yes. Yep, this is inactive.
Okay.
Thank you for asking that. That's a very good point to make clear, and that's because then there's no way to recycle them, so they have no choice except for to be broken down. And when I say inactive on this, I want to make sure and say that it is only slow. It's partially broken. Partially, it's reduced, but it isn't completely nuked.
Okay.
There is still a little bit of Higpert stuff going on, which is why you don't have a whole bunch of other issues, which we're going to talk about with our next disease, when it's completely broken. But first, let's go ahead and mentally review gout. Where are the 3 places that you can make a heck of a lot of uric acid from when this thing, and this thing are malfunctioning in some way? And then what can you do to treat it? All right. So the 2 PRP enzymes can go super fast. And then that gives us a whole bunch of hypoxanthine and guanine and therefore uric acid. Or the recycler, Higpert, is broken and therefore we can't recycle, so we have to make it into uric acid. We treat by trying to turn off xanthine oxidase using allopurinol, which would replace the hypoxanthine saying, hey, try and break me down. And then you can't. All right. So, that was disease number 1. And I'm going to write the diseases over here just because I think that it is easier. So we've done metabolism, then we did salvage, and then ideally there would be, I would draw it out 3 times, but then we wouldn't have time. And then diseases. Next, Lesch I can't spell lesh, but lesh nahan. Nahan. Okay? So, lesh nahan is going to be with our friend Higpert again, except this time it is completely broken. Okay? So, lesh nahan is this guy, Higpert, completely broken. This is why I should have drawn it out twice, because then I don't want too many things all on one, but as you will. So, lesh nahan, if you don't have Higpert, then you are going to end up with lack of these guys, and that is going to end up giving you a lot of issues. Some of them, basically mental issues, and the way that it can manifest, aggression, self-harm. Okay. And that's most of lesh nahan. I'm just verifying one thing really quick. Awesome. Okay. All right. Mental issues, aggression, self-harm. That's what I remember about lesh nahan. All right. Next, after lesh nahan, so we had one here, and then I'll circle here. That's an easy way to do it. But I will circle the diseases on there. Next,
Is the treatment the same?
for Higpert, lesh nahan treatment I don't know about. I would not imagine you do the same thing, because inhibiting breakdown of these guys would not give you, inhibiting breakdown of hypoxanthine and guanine would not give you more IMP and GMP, and therefore it wouldn't take care of the problem. So I would imagine that it wouldn't work to give the same treatment.
Would they be able to ingest Higpert, like barley or?
Oh, try and supplement with functional Higpert?
Yeah.
That, if it was able to make it into the cell, that would fix the problem. I don't know if that's a treatment presently or not. I don't know if we have a good method of delivery, but if we do, then that's a great treatment. All right, Keora. All right, let's mow through the rest of these. Next, we have ADA, which is not the name of the disease, it's the broken enzyme. This one is severe combined immunodeficiencies. SCID. All right, so this is the one where you can't get your AMP to break down. So you get a ton of AMP/adenine, basically the adenine family is how I'll write that. Adenine family, therefore, which is what 3 dots is. I bless the professor that taught me that. All right.
Me too. Technical. Go ahead.
So much faster. All right. Therefore, inhibits PRP. Inhibits purine synth. Purine synth. So basically, if you have a whole bunch of this, you're going to shut those 2 guys down. If you have a whole bunch of the adenine family, then you're going to shut down these because they sense, hey, we got a ton of purines. It's just that they're all the same kind of purines. You don't have the guanine, but these aren't sensitive to how much of each. They're just sensitive to how much, period. And so if you don't have ADA, then you're going to build up a ton of this, and that means this is all you will have because it shuts down the production of new purines. Make sense? Kosher? Okay. Number 4. Oh, this one's fun. Okay. No one has probably seen this except for maybe like 2 people in the room. But how many of you have seen the really old, and if you add nostalgia, it might have been called Well Done, but if you don't add nostalgia, it's definitely not a good movie. But it's called The Black Cauldron, and it's the cartoon version of The Black Cauldron. Thank you. Thank you. All right. So, I am the other one in the room. So, there is a little Elmo-looking creature that is not Elmo, like the Muppet, okay? And his name is Gurky, okay? Gurgy. And he speaks like this. Gurgy, Gurgy, like food. Gurgy, food. Gurgy's excited.
Like
all right. Smeagol. Yeah, all right. So this is called von Gerke's. Von Gerke's disease. And it is right here. And if you kind of like make glucose the head, and you made down here the feet, then that's the throat right there, right? And that's why Gerke speaks so weird. Gerke has long throat. All right.
That sounds like Precious.
That's what I said.
Oh, we could have so much
You
fun.
should ask questions in class like
Preston.
We're running out of time. We're running out of time.
Okay, so von Gerke's, basically, if you can't, so it's going up that is broken. So you can't make, gluconeogenesis doesn't work. And so anything that you are trying to make gluconeogenesis is going to go either to Glyco Genesis or to PRPP. And PRPP is going to end up making purines. And so it just diverts it to the side because it has to go somewhere. Because all the regulation is saying go up, go up, go up. It can't go back down, therefore it has to go to the side. Make sense? Okay. And so von Gerke's is you get a whole bunch of this stuff going on, and you end up having excess purines. And that's because this enzyme right here is broken. The phosphatase, glucose 6-phosphatase, I think. All right. Then our fifth disease was the what was our fifth disease? Oh, right. Okay. So fifth disease is when you don't have xanthine oxidase working. So if you don't have xanthine oxidase, you're going to build up a whole bunch of hypoxanthine and guanine and back up the whole system, and you're going to have these in the blood. It's just called xanthinuria. So you see xanthine and hypoxanthine in particular. Those are the ones we can pick out because they shouldn't be in the blood. There's also guanine in the blood, but that's always there, so it's not the one that we named it after. But hypoxanthine and xanthine, when this enzyme's not working very well, you get more of these two. You still get some xanthine because it works a little bit, but you get hypoxanthine and xanthine built up in the blood, gets filtered out of the blood into the urine, and then you can see it in the urine as well as the blood. Okay? All right, last thing that I will say is that all of these are X-linked recessive except for this guy is autosomal, ADA, which is SCID, so I'll go ahead and this guy and this guy. Xanthinuria. Xanthinuria. Those two are autosomal. The rest of them are X-linked. All right. Any last questions?
That was so helpful.
Yes.
Glad to hear it. Oh, so autosomal recessive, in this case, it's an inheritance pattern. You're going to learn more about it in next quarter, but basically you have 23 different chromosomes where you carry genetic material, right? And so 22 of those chromosomes are generic, I guess you could say. They're non-sex chromosomes. They don't have anything to do with your gender. And then you have one pair, the 23rd pair, that are your sex-linked chromosomes. It's like you either have an X or you have a Y. Well, I mean, you're going to have an X, but you have two X's or an X and a Y. And then if you have an X and a Y, you end up male. If you have an X and X, then you're genetically female. And so when it's called an X-linked, dominant or recessive, an X-linked disease, it means there's something wrong with the X chromosome. Okay? Autosomal means all the rest of the guys. Any of them could have the issue. So we just divide it between the sex chromosomes, 23, or somewhere else. And that's because it really affects who will get the disease. Because my next class was actually canceled. Do you have a class right after this? Mm-hmm. Okay, so I do have some extra time today. So this is actually something that will be helpful for medical genetics next time. So, I mean, like next quarter. So let's go ahead and get into this. But who cares? They might care too. So, X-linked. I want more board. Goodbye. So if you had someone who had a good X and a bad X, this would be a female, and they can use the good X DNA to make all the proteins the right way. Whereas if you were a guy and you had a good Y and you only had a bad X, you don't get a second chance to make those X proteins the right way. And so this is why it makes such a big difference. A guy gets one chance at having a functional X chromosome. And so guys will get X-linked diseases way more often than females, because females get two chances. Mm-hmm. So, yeah. So these three, Gout, Leschnahan, and von Gerkes, are X-linked. And then SCID and xanthinuria are autosomal, which means these three are seen more commonly in men, Gout, Leschnahan, von Gerkes, and then SCID and xanthinuria are probably more evenly split between men and women. All right? Any last questions?
I lost the last disease.
The last disease is xanthinuria. So you get xanthine oxidase broken.
This is number 5. We have 6, right? Or no?
I only saw 5 diseases that were discussed outside. She had a table that I think had one more, but I didn't see it in the slides, so I was like, shuffled it under the carpet. Okay, awesome.
Thanks, Taylor.
My pleasure.
Have a great weekend.
You too.